Okay, this is gene expression. In the green book, this is actually going to be at the beginning of chapter 11, although we're continuing it as part of chapter 10 here. And this is how genes are expressed. Most genes don't stay on. In fact, the default setting for a gene is off. And that way you aren't wasting, the organism isn't wasting energy producing proteins it doesn't need. So what gene expression does is it's going to turn the DNA into a working protein, and we're either going to have the gene on or we're going to have it off. And again, the default here is usually off. And this is controlled by proteins. Now, in prokaryotes, not in eukaryotes, but in prokaryotes, there's something called operons. And operons are a cluster of a gene with a similar function and a promoter. And you'll see a diagram of this next. They can be repressors or activators. A repressor is going to stop the transcription and translation. The activators are going to start it. And these will be controlled by things in the environment. So one of these operons is called the lac operon, and it's going to produce an enzyme that helps break down lactose, an enzyme called lactase. Okay, well, the E. coli, this is in E. coli. E. coli live in your gut. They are a bacteria, so they're a prokaryote. They use operons, but they only want to make lactase when you have had a nice glass of milk and there's a bunch of lactose that they are bathed in. Otherwise, it would be a waste of their energy. So what they have is in front of the genes to help digest lactose. So here you have 3 genes that are going to digest lactose. All right, here they are if they've been made. Here are your enzymes that go digest lactose. Well, we don't want these being read, right? We want our RNA polymerase not to attach to this unless lactose is present. So the way the operon works is in front of it, where the RNA polymerase would normally bind here at the promoter and start working its way down, we have a repressor. It's a protein that physically blocks the RNA polymerase. It cannot bind here, and it cannot move down the DNA and transcribe it. And this is great for E. coli because they don't want to waste time and energy making these enzymes when there's no lactose. But let's say you have a nice big glass of milk and these E. coli are now bathed in lactose and they want to be able to digest it. What happens is, here's the lactose, so they actually use what they are going to digest. There's enough of it around in the cell that it comes here and it will bind to this repressor and cause it to fall off. It'll change its shape ever so slightly and it will inactivate this repressor. And that will allow RNA polymerase to bind, to go ahead and transcribe and then translate these enzymes that will then go digest the lactose. And once they have digested enough lactose, and these are going to rebind on. Once the lactose is gone, they will go find their spot and rebind right here on the operator and prevent the RNA polymerase from translating that DNA strand, transcribing the DNA strand. Now, not all types of operators are active, only when or inactive when something's bound to them. So for lac operon, if the lactose binds, the operator becomes inactive. Well, there's something called a trip operon, which is a nice example of the other kind. This actually will only bind when tryptophan is present. And the reason is the genes down here, the genes here actually make tryptophan. The E. coli is capable of making its own tryptophan. But why bother doing that if tryptophan's present, right? So if tryptophan is in the system, it's going to block the RNA polymerase. It will not be able to bind. And as soon as it runs out of tryptophan, there's no tryptophan present, this will fall off. It will be an inactive repressor, and RNA polymerase will come bind here, and it'll be able to make its own tryptophan. tryptophan. So you can have either kind of repressor, one that needs it bound, like this one needs it bound, or one that will inactivate the repressor when it binds, like the lac operon. Eukaryotes are different, and we don't have operons for one thing. And when we look at eukaryotes, there's one thing called differentiation. Our cells differentiate. What that means is you start with stem cells, but eventually, as we develop, they're going to turn into muscle cells, pancreas cells, whatever. Same thing with carrots. You know, you're going to end up with root cells, stem, leaf, all that's going to differentiate. And so a muscle cell obviously doesn't need to make the same kind of protein as a blood cell, and it would in fact be a waste of energy to do that. The chromosomes in eukaryotes actually get to control the genetic expression. So in some examples here, you have muscle, you have pancreas cells and white blood cells, and then you have some genes, for example, for glycolysis. Well, all of these require energy, so they all have glycolysis turned on. But for muscle contraction proteins, guess what? Only the muscle needs that. The glucagon is only needed in the pancreas, and insulin is turned off except for in the beta cells of the pancreas. Hemoglobin, that's what helps carry oxygen in your blood, and it's your red blood cells that do it, so it's turned on there. But notice how many offs there are. Again, off is the normal default setting. We only have things turned on when we need them. So in eukaryotic systems like us, the DNA packing is really what helps regulate gene expression. Your DNA is wound around histone proteins, and it's wound and wound and wound and wound. And in fact, if you stretched out the DNA in one cell, it would be like 4 centimeters long. Bigger than the cell, but it's tightly wound, and then as it winds around the histones and folds, it forms these little bead-like structures called nucleosomes. Now, what's interesting is in female mammals, one X chromosome is inactive in each cell, and that's actually random, and this is an extreme example of DNA packing, right? This happens as the cat's developing and one X chromosome is turned off. Well, what can happen with female cats, they have 2 X's, obviously. With their color, if you have the orange fur activated in one location and the black fur is activated right next to it, so you can actually see some spots on the cat where you can tell it was right here, the orange was inactivated, and as those daughter cells reproduced and the cat developed into a full-size cat, you have an area of black. You can also see it right on the nose, where this was clearly a patch that had the black inactivated. So this tortoiseshell is in female cats, where they have 2 X's. Transcription factors in eukaryotes also help with regulating gene expression. So what you can have are something called enhancers. And the way this works is for a gene to be expressed, for it to be transcribed and translated, it really needs these transcription factor proteins, right? So these things come in, and what will happen is in front, here's the gene, over here, way off to the right is the gene that we want to express. Here's the promoter, that's going to be where we're going to have our RNA polymerase bind and go do this gene. But way out here, you'll have these enhancers. You go, okay, well, they're way out in front. Well, what they do is they will bind to these transcription factors, physically bend over, and help that RNA polymerase get to where it needs to go, and then it can start transcription. So it's a way of regulating the transcription. If these factors are not present, then this gene will not be expressed. So, like I said, eukaryotes, we don't have operons. We are controlled by transcription factors. And again, this is the same diagram that you saw in the previous slide, where this binds and helps the RNA with transcription. There is another thing that can be done, and it's not on this diagram, with repressors, where they bend the silencer region near the gene, and it prevents the RNA polymerase from binding to the promoter. So not only do we have enhancers that can help turn it on, we have repressors that will be up in front of the gene as well that will help turn it off. And then after transcription, so we can regulate this stuff. After transcription, your gene gets spliced. And remember I said in the last video there were introns and exons, and you can remember introns are like interruptons. They're the ones that get spliced out and removed. And the exons are part of the actual gene. Well, depending on how you slice the DNA, how you slice it up, you can get two different forms of messenger RNA depending on which ones you cut out and which ones you leave in. So that's another cool way of regulating our gene expressions. And humans, they have found over 100 genes that will be spliced in two different ways. Okay, so even after translation, now we have made the protein. We can still regulate it. For example, some proteins degrade fairly quickly. Some stay around for a while. Hemoglobin, which is used in your red blood cells, stays active for a full month. Another way we can help regulate, besides just the life of the protein, is it may need to be active, it may need to be cleaved, it may need to be cut in some way to activate it. So you can see over here in this folded polypeptide chain that it's not going to form the active form of insulin until it has been cut. So that's another form of regulation. This can sit there and wait to be activated. And then some proteins are digested after a certain period of time. And so obviously that will deactivate them. They won't be working anymore. So that's another form of regulation. All right, so this kind of sums up all the places we can regulate our DNA. So we're going to start at the top. When it's in the nucleus, the packing, the actual physical winding of the DNA, if it doesn't get unwound, it doesn't get transcribed. So the physical packing is a form of regulation. And then at transcription, you have the different forms of splicing. You also have the addition of the caps and the tail and how long these are. And then once it goes into the cytoplasm here, how quickly the messenger RNA is degraded, right? If it sits there, it'll keep making protein and protein and protein, but if we break it down fairly quickly, it'll stop. And then once the polypeptide chain is made, we can continue to regulate it by either activating it if it needs to be cleaved or modified in some way. And at the very, very end of the line, when that protein has been activated, we can degrade it. We can break it down when it has done its job, and that's the last step.